literally mean above the genetics. Epigenetics are those traits which are due to the stable and inherited changes which are in the chromosomes, but these changes are not in the sequence of the DNA. There are different causes of the epigenetics: DNA methylation, histone modifications, as well as microRNAs. These are the three important causes of the epigenetics. Cytosine methylation is usually that a methyl group is attached at the carbon number 5 of the cytosine. This is the structure of the cytosine. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4 and carbon number 5. So at carbon number 5, a methyl group CH3 group is attached. This is the conversion of cytosine into methylated cytosine. We can see here that cytosine is methylated. Again here that cytosine is methylated. DNA methylation or the cytosine methylation is epigenetics, but it is not the changes in the sequence of the DNA. These can be considered as modifications to the DNA sequence or modifications to the chromosome structure, but actually these changes are not in the sequence of the DNA. There are two primary mechanisms by which epigenetics occur. The first phenomena is called as cytosine methylation, while the second phenomena is called as modifications of the histones. So these are two primary mechanisms by which epigenetics occurs. Molecular mechanism, that how methylation occurs in the DNA sequence and how there are post-translational modifications of the histones. Here we can see that this is the structure of the chromosome. Chromosome consists of chromatin fiber, and chromatin fiber is built up of the building blocks which are called as nucleosomes. So chromatids, they are made up of nucleosomes. So within the nucleosome there are histones. There are 8 molecules of the histone within each nucleosome, and within these histones there are post-translational modifications that can be in the form of acetylation or in the form of phosphorylation. So these are the common methods by which there are post-translation modifications to the histones, while on the other hand, in case of DNA sequence, this is DNA sequence where we can see that our DNA is methylated. Here is C, while here is G, and C is methylated. A methyl group is attached to the cytosine. So here is methyl group that is attached to the cytosine of the DNA. So there are two basic mechanisms by which epigenetics occur. One is the histone post-translational modifications, and second is the DNA or the cytosine methylations. Here we can also see in detail that how histone modifications occurs. These are the histone molecules 1, 2, 3, 4, and same is on the lower side. So there are total 8 molecules of the histones. There are modifications to these genes. Due to the histone modifications, genes, they can be switched on, and the genes, they can be switched off. That are due to the histone modifications. So there are different histone modifications. We can consider that some of the enzymes, they work as the writers, some of the enzymes, they work as the erasers, and there are some enzymes that work as the readers. Some enzymes, they add histone modifications, so they can be considered as the writers, while those enzymes, they remove the histone modifications, they can be considered as the erasers, while those proteins or those enzymes that enhance the gene expression or block the gene expression, they can be considered as the readers. In addition to two primary mechanisms, there is another mechanism that have been invented, that have been identified, that is called as a small interfering RNA. Small interfering RNA is also responsible for the epigenetic mechanisms. Here we can see that how small interfering RNA works. There is a double-stranded RNA. This double-stranded RNA ultimately converts into siRNA duplex, and as a result of certain reactions, this siRNA is converted into single-stranded RNA. That single-stranded RNA forms a complex with the messenger RNA. So when this siRNA forms the complex with the messenger RNA, that block the working of the messenger RNA or the translation of the messenger RNA and silence that messenger RNA, so the gene no more will be functional. So small interfering RNA is also a phenomena that is behind the epigenetics. If we do the comparison between the genetics and the epigenetics, in case of genetics, we can see this is genetics and this is epigenetics. In case of genetics, there are the mutations, and mutations are on the sequence of DNA. So they occur, or they are the changes in sequence of DNA. While on the other hand, the epigenetics, in case of epigenetics, these are called as alterations. Alterations, they are not the change in sequence of DNA. Instead, they are some modifications. Modifications on the histones or the modifications on the cytosine, which is present in the DNA sequence. In case of genetics, that is inherited, present in the germline cells, and it is in the species. While in case of epigenetics, it is stable in the soma cells, and there is variability between the cells and between individual to individual. So we can conclude that in case of genetics, there are the mutations, or there is a variation in the sequence of the DNA, while in case of epigenetics, there are certain alterations to the sequence of DNA, that is addition of the methyl group on the cytosine, or it can be by acetylation or phosphorylation. So we can conclude that epigenetics is a phenomena, that is certain changes occur in the structure of the chromosomes, while in case of genetics, the changes that are in the sequence of the DNA. Epigenetics is a stable and heritable phenomena, but that is not the change in the sequence of the DNA.